Yeah, I'm really excited to give this talk actually. I really like this whole organization. We've been a part of it. My lab has been a part of it for a few years now. We actually came to this space before it was renovated, or during the renovation, I guess. And we were in some like room with a bunch of boxes. And now this is amazing. I don't like I will look that way instead of at all of you. It's very nice. So I started at the University of Chicago in 2020. Our lab has been around for about 4.5 years. And what I'm going to tell you about today is very much a work in progress, but it's a narrative that has been built over a few papers that we've put out. And it's about getting towards the generative framework of emergent phenomena. So I want to start by like defining terms. And these terms are terms that are used a lot in the literature, but don't really mean much when you dig into them. So I'm going to caveat everything that I say with, I am defining these terms. This is what they mean to me. Please don't burn me at the stake for using complexity and emergence in the ways that I'm going to use. Just know that they're subjective. So we think of complexity and emergence as interrelated words to describe systems that intuitively are hard to understand for us. So what do we mean by this? Complex systems, to me, mean that they're comprised of many, many, many parts, and those parts interact together to create some macro functional property of the whole. That does not mean that, that to me does not mean the same thing as emergence, because we can write down wiring diagrams potentially of complex systems. So what you're seeing there is a Boeing, I don't know if it's a Boeing 747, but it's a plane that has a whole bunch of parts. And to the degree that we can write down the instruction manual, we can predict how long it's going to take that plane to go from point A to point B, almost to like the minute precision. But underneath the class of complex systems exist emergent systems. And what I'm showing you here are a bunch of emergent systems. Notably, they're all biological systems. But the way that we in the lab disentangle these two words is to say that emergent systems cannot be understood by the sum of the parts. What that means effectively is that there is no wiring diagram that we can write down right now that can tell us how to put amino acids together to create folded and functional proteins, or how to put genes together to create functional cells, or how to put microbes or species together to create collective ecosystems that do something like foraging or schooling, or in the case of your gut, controlling the metabolism and physiology of humans. And if we dig into that statement, it's a little, oh, go ahead. I think that's not a bad way of saying it. Yeah. Yeah, I think that's not a bad way of saying it. I think there's, not understand means something mathematically, meaning that I think that the contra is true. We can understand complex systems that have wiring diagrams. We may be able to understand, and this is what we'll eventually show, at least how to build complex systems that are emergent for which we have no wiring diagram, no forward model. But it's a reasonable way of thinking about it. So the question is why? Why can't we understand this? In the systems that I just showed, they all come from a process that we're all familiar with, the evolutionary process, which in some conceptual way can be wrapped under the path integral of selection and variation over time. Okay, so you have systems that undergo experiential processes of iterative selection and variation. The reason I say experiential is because there's no deterministic way of knowing what's going to happen in the future. You have a selection event that creates variation, that selects populations, they vary, then they get selected for again, so on and so forth. And from this comes remarkably ordered structures. Protein cells, tissues, ecosystems that are all comprised of many, many parts that interact together to create the whole. The interesting thing about these kinds of systems is that these emergent qualities aren't nice to haves. So unlike something like birds flocking, where sometimes they can just dance because we don't really understand why. Here, these emergent qualities are critically important. The fold of proteins is very important to execute their function. The organization of genomes is very important to ensure the fitness of the organism. The organization of tissues is very important to create the collective phenomenon of the tissue, and the same thing with microbiotas. So what I mean, and this goes back to the question, what do I mean by emergent systems? Well, we have no insight into the constructive processes of these systems. We have no forward model that tells us, given a list of parts, how do I create a functional whole? This is very different than the Boeing 747 case, where you do have a list of parts and you can create a functional whole because you have this box in the middle that tells you how to put everything together. Why is this so hard? The critical reason is because the interactions between these parts give rise to properties that are intrinsically unpredictable. So unlike linear systems theory, where you put parts together and you have a theoretical understanding of what will ultimately come of putting those parts together, here, this is not the case. You can put two microbes together and you'll get something that is completely not additive. You have a designed protein, and for whatever reason, it doesn't work until it works for reasons that are not known. So this problem is a problem that pervades all of biological systems and is one that is not just theoretical. When we try to build synthetic biological systems, we run into this problem almost ad nauseum. Now, there have been historically strategies to deal with this issue. So I'm going to talk about two strategies. There may be more, but certainly there are two. For a long time, we have thought that we can inherit systems from nature and reduce them. So what do we mean by reduce? We mean take them apart, study their parts individually, put them back together, try to understand the whole. Or inherit them, perturb them, so tickle them in some way, and try to understand the relative sensitivity of each of the parts, because we have some underlying hypothesis that the parts are heterogeneously important to understand the whole. So I'm showing you here at the scale of proteins, genomes, tissues, and ecosystems, there have, since I would say probably as of 10 years ago, there were technologies that were created specifically for this purpose, and in a very sort of seemingly comprehensive way. At the scale of proteins, there was ways that you could mutate every single amino acid of the protein to every other amino acid and get what is called a deep mutational scan done on your protein to understand how relatively important any part is to the whole. At the scale of ecosystems, there was a technology called transposon mutagenesis, where you could specifically target microbes in a microbiome to get rid of them and see what the function of the microbiota was. But it should be clear, and I think many of the people who have created these technologies noted this failure mode, that this kind of way of thinking about such systems does not work. And the reason it does not work is because of the possible synergies between parts, or the non-additivities between parts. So what I'm showing you here is a paper that was actually from the 80s from Alan Fersht that talked about this problem at the scale of proteins. This is a seminal paper because it introduced the idea of thermodynamic coupling and how to measure thermodynamic coupling between parts of a system. So imagine you have the wild type system on the left, and you make one mutation and put it to state 1. You make another mutation to make state 2, and then you make the double mutant. So now what you can do is measure the coupling between part 1 and part 2, depending on the non-additive nature of the functional consequence of those mutations. Okay, so this tells you, well, I had to create 4 observations to now look at two-way coupling. You would have to create 8 observations to look at three-way coupling. You would have to create 2 to the n number of observations on that order to understand n-way coupling. So in the limit that you don't know a priori what the pattern of coupling is in these systems, then you lose because of complexity. This combinatorial problem scales as 2 to the n. which makes this a very hard problem to understand no matter how many liquid handling robots one has or how much deep mutational coupling, tripling, quadrupling scans one can make. So enter AI. This has actually been a great use of data because it tried to get around this problem, even though perhaps this has not been the way that it has been couched to all of us, at least at the scale of proteins. The idea was to say, well, one way to get around this combinatorial problem is to just de novo learn statistical structures of what exists. And instead of doing any mutational scanning, just make models. This is now actually the way that protein structure-function relationships are being dissected almost entirely throughout biophysics. The ideas of reductionism having gone out, dated to the past. So what's the idea? Is that you go out into the world, you take what evolution has provided, you create an ensemble of related systems, and then you plug them into a deep learning network or some network that parses probability distributions in a very sophisticated way, and then ask whether that network can generate new systems. And I put new here in quotation marks because actually all this can do is, by definition, can generate the statistical structure of the input. But effectively, what this is asking is, have we learned enough about maybe not the mechanism, but at least the statistical distributions to understand how to create new things? Now, there's a central problem here. It's been remarkably effective. So in 2000, or I think it was last year. Yeah, last year, David Baker and Demis Hassabis and John Jumper won the Nobel Prize for this very idea, which is to say that we can create statistical representations of proteins, and they're very good at then generating proteins. But there's really no understanding of constraints here. So what do I mean by this? You create a bunch of proteins, and you get a 100 amino acid sequence, and you have no idea which of those amino acids is actually more important or less important than the others for the functional consequence of what you're trying to create. This becomes not only a practical problem, but also a theoretical one, because if we don't understand constraints, now we lose the risk of being able to understand laws and symmetries in nature. We have no ways of comparing what we're learning or understanding what's invariant across all of the ensemble of stuff either that we model statistically or create to then port into the lab. So our lab has been interested in the problem of trying to infer constraints on emergent systems now for a while, since we started. This was the idea of the lab to begin with. And what I'm going to show you today is a few papers, go through a few papers that have shown that we can do this, and then the work of Bipul sitting here showing that we can translate this learning into a new generative class that is based off of constraint-based learning rather than deep learning. Okay. So we focused, when we tried to understand this problem, we focused on bacterial genomes as a model system. And the question is, why did we focus on bacterial genomes? There was a lot known about bacterial genomes. So we have good ground truth about some of the pathways and the protein-protein interactions and the relative contribution of proteins to fitness. We also have the assay capabilities of growing these bugs, of mutating them, of getting knockout libraries. So it was a particularly good assay system to try to think about how emergent properties manifest from the layer of parts, where the parts are proteins, to the fitness of the organism through what we think is the structure that biology has created, which is that if you have amino acids that create proteins, those proteins create protein complexes, those complexes create pathways, and the pathways ultimately create metapathways and affect the fitness of the organism. So we had a good prior to think about, well, if we're learning emergent constraints statistically, then we can at least compare it to what people know. I should say that's different than at the scale of proteins, where you don't actually know if there's a hierarchical prior. You also don't know this at the scale of ecosystems. You also don't know this at the scale of tissues, right? At those other scales of biology, there is no ground truth, whereas here you have things like the KEGG hierarchy that tells you, here are proteins, here are complexes, here are pathways, here are metapathways. Okay. So what did we do? Um, yeah, okay, this I just said. So here is our strategy. So similar to this schematic, where we go to the world, we take in a whole bunch of sequences, we did the same thing. Okay, so UniProt has a reference proteome database of 7,047 organisms that have all been whole genome sequenced. None of these are redundant organisms. They all represent independent strains of bacteria taken from metagenomic sequencing. Now, you can annotate any one of these bacteria using what are called orthologous gene groups. You can think of this as gene content that's alignable. So any orthologous gene group is related to another one through sequence, not through function. So inferred sequence, conserved structural motifs from a primary amino acid sequence perspective. But the way to read this matrix on the left, which is the matrix that we created, is every row here is a bacterial proteome, and every column here is a COG, or an orthologous gene group, where the number of genetic elements is reflected in the matrix. Okay, so like Pedobacter has one COG 1283 in its proteome. No. So if you take a genome sequence, what do you have at the end of a genome sequence? You have like nucleotides. You can translate that into proteins, right? And then those proteins belong. There's an existing hierarchical categorization called COGS, orthologous gene group categorization. How does it work? It looks at conserved motifs across different protein families and groups those protein families together. So technically it's off the Blossom matrix. So you make an alignment, you look at what's conserved, you put that into a category. Yeah. Each gene has multiple COGs. Yeah. I don't think I have an example of it, but yeah, you could imagine one gene has like COG1, COG2, COG3. Not all proteins have COGs associated with them, so it's entirely possible that we've missed some of the proteome. But what I'll show you is that we were still able to recreate phylogenetic relationships, meaning that at the very least we captured some essence of the proteome. Given the complexity, 10,177, you can go and look, at least in E. coli, we have a pretty good coverage. I think it was like 94%. That being said, not all bacteria are studied to the same degree as E. coli. So make an assumption. Yeah. Okay, so here is the question. To what degree does this matrix reflect the information that I'm showing you on the right? Okay, what is the information on the right? It's a veritable hierarchy of biological information. So at the top, what you're seeing is phylogeny, phylum, class, order, family, genus. Okay, and then what we were able to leverage were databases that exist about non-interacting or directly interacting proteins. So the string database, the Go database, these are non-interacting protein databases. And then Coev Plus is a computational method. Forget that for a second. But the PDB and EcoPsych are databases that tell you whether two proteins directly interact with each other. Okay. So the question was, does this matrix encode that information? So we performed, to look at the statistical structure of the matrix, we performed SVD on the matrix, and this is what we found. So there's the eigenspectrum. It looks like an eigenspectrum. And the top modes of variance told you something on the order of 20 to 30% variance. So if you look at the top modes, which are shown on the bottom there, each dot there is a projection of one of the 7,047 bacteria onto those modes. And what you can see is that they split along pretty much phylogenetic terms. Okay, so yeah. Maybe. I mean, it's not functionally aware. It's not de novo phylogenetically aware, but you could imagine that the conservation pattern probably has latent within it phylogenetic information. Yeah, it's totally reasonable in the same way that the genome does, right? So it's difficult to disentangle the two from each other. I mean, one thing worth noting here, one wonders how much worth there is in parameterizing all of this by a completely orthogonal way of thinking about things like the first letter of the name of the bacteria or something like this. I don't know. Yeah. So there's probably some tautology here. Nevertheless, the way we get these genomes and we decompose them is through their genome sequences. This is a reasonable proxy for them. So if you look at the statistical structure, it's not surprising. People have shown this before, that the top modes of variance tell you about phylogenetic distinctions. Here is the interesting thing, though. What happens if you look at deeper components? Okay, and the deeper components here, I don't mean like PC 5 through 15. I mean all the way down to this hump right there. Okay, so here's what we found. We actually found a hierarchy as a function of how deep one goes in the spectrum, out to component 3000, which held less than 10 to the minus 5 amount of variance. Okay, so what you're seeing on the graph at the bottom that has the rainbow plot is the relative mutual information of each of the categories on the left with respect to where you are in the eigenspectrum on the x-axis. And what we found was that the top modes of variance told us about an ordered hierarchy that specified phylum, then class, then order, then family, then genus. Then below that, what we found was that the information about indirect interactions lived here. And then the direction about the information about direct interactions lived at the bottom. Okay? So this was a result number one, that the eigenspectrum isn't simply just signal to noise, which is how I was taught it was. It actually is a spectrum of information that contains scales of organization, maybe from local to global. Okay? So the second thing that we asked was, could we use the information in the eigenspectrum to de novo derive emergent organization statistically? And I'll just show one example of this. We showed this more generally in the published paper in eLife a few years ago, but here what I'm showing you is the flagellum. So this is a very well-studied bacterial set of components. The flagellum is the part of the bacterium that enables bacteria to move towards a target, and it's made of a whole bunch of proteins. It's also nice because it's been studied to death. Like 40 years of research has been done on this. Many people have done modeling. Many people have done knockouts, double knockouts, triple knockouts, good annotation of pathways, etc. And so we have a good hierarchy to compare against with respect to ground truth. And so the question was, could we use the eigenspectrum to recreate the flagellum and directed motility in a more reasonable way? Okay, in a more statistical way. And the short answer was yes, we did. So here's how we did this. I'm going to gloss over details for the sake of time, but on the left, what you're seeing is that rainbow plot. And you can think of it as if you start at the bottom of the eigenspectrum and move up, what do we see? So at the very bottom of the eigenspectrum, what we saw were all the proteins just independently varying and no covariation with each other. Okay? And then when we moved up, and I labeled this as the bottom layer, what did we see? Well, every node that you see here is an individual protein in the E. coli genome. And every edge that you see is those two proteins statistically covary with each other in a non-random way in the window of the bottom layer. So in this case, it would be in the window of the direct interactions around here. Okay? Okay. And so if you look at this and you do gene set enrichment analysis, what you see is these modules are not random. They are the individual parts of the flagellum, right? You see that there's a module that is named flagellar rotation. There's another module for flagellar basal body. There's another module for flagellar LNP rings, for the flagellar for the flagellar body, the filament, so on and so forth. You also see that there are modules for signal transduction, chemotransmission, and chemoreception. Okay? If you then move up the eigenspectrum and go to the middle part of it, what you see is those modules coalesce into cooperative units. Three modules, 1, 2, 3, and then 4, right? What are those? Those are also non-random. That's the integration of each of the parts at the bottom into macromolecular complexes, the flagellum, and then the signal transduction machinery for chemotaxis signaling and cyclic di-GMP signaling. And then when you go to the very top, everything coalesces into one unit, which is directed motility, right? So what this showed us was that just by looking at the statistical structure of variance, not only could we parse different pieces of scale, but those scales were related to each other. That if you marched from the eigenspectrum from bottom to the top, where bottom is like percent variance that we really never look at in biology, what we see is individual parts to collective whole. And what I'm showing you here is a mapping between the statistical hierarchy that we found and the KEGG hierarchy that was known in literature. Yeah?
What happens if you titrate the amount? So,
Yeah.
new experiments on new things where you don't have as much data. What can you expect to learn?
It's a great question. Can I get back to you? Yeah, I'll come back at the end of the talk. I think we'll answer this. Yeah, go ahead.
What these lines actually mean in the bottom layer? So
The lines. You mean the edges?
Sorry, yeah.
The red lines?
Yeah, between the nodes.
Yeah, okay, yeah, sure. Yeah, we can go into the like. Yeah. There's a null model of what are called. Okay, so how do we actually compute a line? In this case, we take windows of the eigenspectrum. So let's say left singular vectors 1 through x. In this case, it was 1 through 5, 6 through 10, so on and so forth. Then we compute what's called a spectral correlation. Okay, so what that means is if you look at two proteins, you can take their projections onto LS, left singular vector 1 through 5. Now imagine you've subset that matrix and you've got two proteins by left singular vector 1 through 5. You can compute that correlation, right?
The correlation.
Well, it's not the correlation between two vectors. It's the correlation of the left singular vector projections across the window of left singular vectors. Okay? So then that gives you a correlation value that gives you a proxy for statistical interaction within a window of the eigenspectrum. Right? And then you can ask if that's over random. That's it. Yeah. Okay. Okay, so key result number 2 was that information from the eigenspectrum alone provides a wiring diagram for these for emergent systems, and we showed this more generally in that paper. And what the last thing I'll say before getting into kind of the newer work that our lab has done is why does the eigenspectrum tell us about emergent hierarchies? It was not clear to us that this was a reasonable property, and the other thing was that everything that we extracted from this paper required us to know information. It required us to know the phylogeny. It required us to know ground truth. So was there a more data-driven way we could use the eigenspectrum and interrogate whether hierarchies are latent within structures? So this was a paper that was written in 2025 by Ben, who's sitting here, that showed that the eigenspectrum, by mathematical definition, creates hierarchical levels of relatedness. Okay, so you can dig into that paper for the mathematical detail more, but what I'm showing you here is an alignment of more of a simulation of a phylogenetic tree where you have a hierarchical process that creates diversity of species A through H. And I'll try to get through this a little fast so that I can actually get to the new stuff. But what effectively we were trying to ask here was if we take an alignment of extant diversity and perform eigendecomposition, what is the information content that the eigenspectrum tells us about that extant diversity? Okay, so what you're seeing on the right here is a decomposition of that alignment, right? So that alignment has latent within it some structure about the generative process, but we don't know what it is. And what we get out from here are principal components 1 through 8, because there are 8 species that we are comparing against each other. So this is what we find, that if you look at the projection of each one of these species onto principal components 1 through 8, what you see is a interesting pattern. That is that at principal component 1, all of the species are not distinguishable from each other. But at principal component 8, what you see are more fine-grained distinctions between A versus B, C versus D, for instance. In principal components 5 and 6, you see E versus F, G versus H. And then in principal components 2 through 4, what you see are A through D versus E through H. Okay, so if you now convert this idea into a mathematical formalism that we call spectral distance, which is how close are two species to each other as one moves through the eigenspectrum, what you see is that the non-zero spectral distances at the top of the eigenspectrum define global differences between all of the species. But the spectral distances at the bottom of the eigenspectrum tell you about very fine-grained differences between those species. So we created a way to convert the eigenspectrum into an inferred tree of relationships that we call spectral inference. Okay? Yeah.
Data can be partially observed, for example, because we don't actually know, right, the true phylogenetic relationship. There's a lot of uncertainty.
Yeah.
For example, what if I have data that's going from like multiple different like wires or I guess like hierarchies?
Yeah. Well, so the only I'll try to explain two things about the question you asked. There's reality, and then there's like what we actually try to do, and then there's ways we can screw up the generative process to ask whether we're, if we have multiple hierarchies in there, like what happens, right? So in this paper, what we showed was that in reality, if we do this off of the same matrix that I had showed you before for the entire kingdom of bacteria, you get a better defined phylogenetic tree. And that phylogenetic tree gets you down to subspecies level variation, okay? Relative to something like phymol, raxmol, and any of these, what is it, GTDB, these kind of classification schemes that actually corrupt the inference process, okay? So that's like reality. The second thing is we did do some kind of kooky experiments where we tried to inject convergence, like evolutionary convergence, where you have the same, the same thing happens at the end, but it comes from two different processes. Okay, so here's the critical thing about that. We didn't load that in this, example for ease of just explanation, but what is convergence? It's effectively epistasis between two parts. So there's some contingency that you have to disentangle. But the nice part about that, and we showed in this paper, is that the middle principal components directly disentangle that contingency. So you can figure out what the epistasis is because of the hierarchical nature of you need to do this first and then you get these two solutions versus you need to do another thing first to get the same two solutions. Your point about incompleteness is a very good one. So only until recently did we have complete genomes of bacteria. So now we have the ability to do this with bacteria. But for other things, like if we tried to do this on, let's say, cellular systems, right? What would we need? Something like there's a paper that we have in revision that has done this on cellular systems where we have the whole transcriptome. What we show in that paper is that you can subset the transcriptome to just, multiplex IF patterns that you can get, and you can recapitulate the biology. But critically, we didn't go into that thinking, oh, let's subset the information. We wanted the entire, genome or transcriptome. So it's a question about how well you can parameterize the system to begin with. I think now technologies enable this, but it has not always been the case. Did that answer your question? Okay, good. So what was the result from this paper? It was that the eigenspectrum encodes a tree. revealing scales of organization that are related to each other hierarchically. Okay? So I still haven't got to the title of the talk. Now I finally do. So the question is, could we use what we've learned as a hypothesis for a generative framework of emergence? So what is the idea here? You take an ensemble of related systems, infer scales of organization, and then here was kind of the assumption that Bippel made that was, I would say, an egregious assumption, which is let's only train these models based on the interactions between sequential scales of organization. Okay? So here's what I mean by this. Okay, so this is him. What is the intuition? It's that if you look at this eigenspectrum, you have no, just like what was just said, you have no understanding of the true generative process. But what is this telling us, at least statistically? It's telling us that there are different scales of entropic organization that you can extract from the eigenspectrum. At the top, what you're seeing are slow varying, low entropy pieces of the system. In the middle, what you're seeing is faster varying, medium entropy pieces of the system. And at the bottom, what you're seeing are very fast timescale fluctuations that create high entropy organization of the system. Were you going to ask a question? Yeah.
Yes. Let's get something for that.
Yeah.
You do PCA on a noisy column, right?
Yeah. Structure.
You will also be able to
very good.
Signs that in one of these times you get signs
yeah.
Using the high frequency, you get the Fourier specifically.
Yeah.
And if your notion of hierarchy is orthogonality along the subspace spanned by the rest of the definition, then you can build a tree defined trivially by this.
Yeah.
So I guess
can
you give us a more precise statement about what you mean by this hierarchy attaching to the hierarchy that might exist in data as opposed to those for noise.
We'll get there. Yeah, we'll get there. Is a very good point. So what was just brought up is if you perform PCA on noise, you get something that looks very similar to this kind of the scaling properties of what I showed you with these eigenspectra, right? There is an outstanding question still that I think we're working on, which is what exactly is the difference between noise and this? But part of this was to, ultimately going to the generative aspect was to try to see whether or not we could start disentangling this idea a little more. Because the idea was that if we could generate something, we'll know if that's noise or not. But yeah, I'll get to your point in just a second. What does the JIJ actually look like given these constraints? Okay, but this is the intuition behind the idea of imposing a constraint on how to train these models from the statistical structure of what we would learn. Okay? So you can think of this as a prior, right? I mean, the other way of doing this would be to just take in what you get and create a POTS model and be done with it. This is what has typically been done, and effectively so, right? DCA in protein design did this in 2011, and it worked pretty well. So that's like the alternative to what we're talking about. Okay, so to give you just one more piece of intuition, what does it mean? It means that what we're saying is that the way that these systems can be generated is by instantiating the low entropy stuff first, then building the medium entropy stuff, then building the highest entropy stuff. Okay? What that means is that the lowest entropy parts, the organization that one would infer from the eigenspectrum, does not explicitly talk to the high entropy parts. It talks to the high entropy parts through the medium entropy parts. So there's a serious amount of constraint on the JIJ that you would put on, which would say, I don't want the inferred ordered components of the system to talk to the components of the system that I can see are fluctuating very fast across the ensemble of systems that I've inherited from nature. Okay, so we call this adiabatic entropic coupling because the way that we do this in terms of training is to allow the low entropy organization to effectively cool first, then build upon that to create the medium entropy organization, allow that to cool, and then create the high entropy organization. So very practically, how does this look like? You have a system on the left, system 1 through n, that are described by features 1 through m. The bits of information can be whatever you want them to be, right? alanine through tyrosine, a 1 and 0, species is there or not, whatever it may be. You compute the entropic scales using the eigenspectrum, and then compute the interactions, or in Potts terms, what's called the JIJ, under the adiabatic entropic regime. And then you train the model on just those interactions. And what this ultimately leads to is an architecture that looks like this. A layered architecture where layer 1 talks to layer 2 talks to layer 3, so on and so forth. The input into this becomes your, let's say, noisy system, and then the output should be, if it's a well-trained model, a synthetic system that kind of looks like the input. Okay, and we called this the layered restricted Boltzmann machine, because like a restricted Boltzmann machine, it's trained off of interactions, but it's layered in the sense of the priors that we put on it. Okay, I'm going to take you through 3 cases. The first case being our foray into MNIST, the second case being protein design, and the third case being Jane Austen novel. Yeah, that one's interesting. So to give you some intuition about what we mean by slow varying to fast varying, tell me if any of you don't know what MNIST is, but effectively MNIST is just handwritten digits. It's a digitized form of handwritten digits from 1 through 9. Okay, that's how they look like on the left. And if you think about what slow varying to fast varying means, slow varying stuff is any part of this image that a digit is not written on. So it turns out that there are a lot of just blue pixels no matter what in those images, because no one writes into the bottom right part of this, right? So that's slow varying, right? Fast varying pixels are all the pixels that are in the area where you write, okay? So that's why you see at the fast varying part, we identify this area that kind of looks like a hollowed out 0 as the fast varying pixels. Okay, so we have the same random input, and what I'm showing you here is the evolution of that input into what seems like reasonably handwritten digits through layer 3 of the Boltzmann machine. So what you're seeing through layer 1 is a constraint of taking that random input and then subsetting the variation into the slow varying parts. Then you see, I mean, it's hard for us to really tell what the medium varying parts are, but then at the fast varying parts, what you start seeing are curvatures that look like handwritten digits. And we can find that as we put the same random input into the layered restricted Boltzmann machine, it's not just learning one type of digit, it's learning the phylogenetics of all digits that were input into the machine, right? So you notice that here you're seeing a 9, a 4, an 8, and 6. And what we also noticed was that it's not just memorizing the input either. So you can see down here, I don't really know if this is a 5 or a 6, okay? I mean, this looks very similar to a 9, could also be a 4, right? So it's worth noting that the Hamming distance is not by like what we think of as Hamming distance, 1, 2, 3, 4. It's rather the Hamming distance in this basis of handwritten digits. But it's what's that? These are stochastic outputs. Yeah, yeah. Yeah. I'll get there. Yeah, we didn't, we have this, I think, for this, but we definitely have it for proteins. I'll show this in proteins in just a second. Yeah, yeah. The most notable difference was that you don't get this. Yeah, you tend to memorize the inputs. Cool, right? So to this point, okay, everyone has shown stuff on MNIST, but we wanted to see whether the same type of approach could be general for other types of systems. So we took the case of corsmate mutase. This is an enzyme, so it automatically raises the bar of protein design in some ways because people think that enzyme design is a very hard thing to do. There was a paper written in 2020 from Rama Ranganathan that designed a whole bunch of proteins according to some model. But it doesn't matter. They had good biochemical data for this, so we thought this would be a good model system to use. Corsmate mutase is a conserved enzyme that allows bacteria to grow. That's all we really need to know here. But the matrix here was a number of natural corsmate mutases by positions in the amino acid sequence, where every pixel was an alanine through a tyrosine. So whether or not a specific amino acid was there or not. And so the first thing we did was compute entropic scales. And to give an intuition about low entropy versus high entropy or slow varying versus fast varying here, the lowest entropy scale here corresponded to that protein structure. That probably doesn't mean much to a lot of us, certainly not to me. But what we did note was that if we looked at the amino acid distribution, we saw heavy constraint on what was allowed. And what we also noted was that that heavy constraint was mostly in the hydrophobic core of the protein. This makes complete sense when we think about what is the most conserved parts of proteins. It's usually the hydrophobic core. If we look at the last partition, which is the highest entropy part of the system, what we find are a degeneracy of possible amino acids that are available. And we also find that most of those positions are surface positions in the protein. You can see that on the colored part there. So here's what the JIJ actually looks like. So what we've done here is ordered the parts by whether they belong to the lowest entropy, medium entropy, or highest entropy. So in the top left, what you're seeing is the lowest layer, and on the bottom right, what you're seeing is the highest layer. And what I want you to note here is that the non-zero part of this is really just along the diagonal. But this is a way of visualizing that the model was trained on a layer and then the interaction of that layer with the one right before it. And that's it, right? There is no non-zero term anywhere else on this matrix. This is very different than the full pots. So this goes back to the question that was just asked. The full pots, which is what the Veit and Ranganathan group had created, has this many number of parameters, 4 million parameters, right? And it's because there are non-zero elements in this off-diagonal term. So this is like a fifth of the total JIJ space that one could train on. So does it work? So this is the whole schema. 1 + 2 gives you this layered restricted Boltzmann machine. Okay, what I'm showing you here in the gray distribution is the training data. In the red distribution are randomly created proteins, not according to I mean, they're randomly created just by that. They're not according to any sequence constraints. And what I'm showing you in the green are 600 sequences that were generated and scored according to the layered restricted Boltzmann machine. Okay, so they were created by the layered restricted Boltzmann machine, and you can score them because each layer is just a Hamiltonian. So what you're seeing is that there's direct overlap between the generated sequences and the training data. What we also note is that all of the joint frequencies were of the generated data were preserved. Why is that so interesting, right? Remember what we trained on. We did not train on all of the joint frequencies. We trained on only the sequential layers of joint frequencies. Layer 1 to layer 2, layer 2 to layer 3, so on and so forth. Layer 1 to layer 3, this area had explicitly zeroed out joint frequency distribution. Yet the total distribution of joint frequencies was conserved. This is a criteria that people use in the mean field approximation of plots to have inferred Hamiltonians off of statistical distributions. So we also capture this constraint. Yeah.
Just easier writing.
Yeah. I might get there. Ask the question again if we don't. I think you have an answer to this, but yeah. Full pots, so using a DCA approach gives you these energies. Okay, so the first thing to note is that those energies actually don't correspond to the training data, right? They don't overlap with the training data, whereas the LRBM generated sequences, they do overlap with the training data. Okay, understand that x-axis, though, is the LRBM sequence energy, right? So we could be being like, oh, well, like, so great for us. Okay, so, right, I completely agree. I see a lot of people nodding. They're like, what the hell, man? Okay, we did a functional assay to really show that this was true, right? So you can, this assay was you take these sequences, you put them in E. coli, and you effectively ask if they grow in the background of no phenylalanine or tyrosine being added to the media. So if they have wild-type corazmate mutase, they're going to grow. If they have a knockout, they're not going to grow unless you add phenylalanine or tyrosine. And so if it's a bad enzyme, it's not going to grow. If it's a good enzyme, it will grow. And then you can perform sequencing and do relative enrichment. Go ahead, yeah. I'll get there. Yeah. That's like the very next slide. Yeah. So the point is that when you look at the LRBM energy landscape, effectively, if you're over here, you work. Okay, and this is across a large number of sequences that are large Hamming distance away from each other. So let's just focus on the green for a second. Okay, so these were the generated sequences from the layered restricted Boltzmann machine. What you're seeing on the y-axis is the Hamming distance from training data, and you can see that there are non-trivial. There's a large group of non-trivially far away sequences from the most close sequence in the training data. So it's not like we're just recapitulating the training data. It's also worth noting, though, that if you compare to full pots, which also gets you non-trivial distances from the training data, you shift the energy distribution towards the right. Okay? And now you start getting things that don't work. So the layered restricted Boltzmann machine is what? Is sparser, is creating new protein sequences that work, and those protein sequences are non-trivial Hamming distances away from the training data. Yeah. No, it's not. It's not. It's so this, as you start effectively going above 500, you get stuff that don't work. Right. So you can think of anything here that way doesn't work. Right. And then this way works. Yeah. Okay. And this is just to show that, like, if you put this into AlphaFold, it gives you something that looks like an AlphaFold output. Whether or not one wants to believe this is off a neural network that also has its flaws, but it's just to say that, it's a reasonable proxy for fold. Okay, so good. I have 3 minutes. We've shown that the power of the layered restricted Boltzmann machine for both of these cases. And so what we tried to do is really push the envelope to do something completely out of the box. Okay. And the reason we did this was because we thought, well, look, like everyone thinks of AI as some like language bot right now. Does this work? And it was really just like a shot in the dark, so to speak. But if it did work, it would show really that there's something really underlying the generality of what we've understood and created. So here's what we did. We took 300 sentences from a Jane Austen novel. It was not Pride and Prejudice. I don't know which one it was, but I know it was not that. And we matricized those sentences. So every sentence was described by the presence or absence of a possibility of 120 words. And we did the same thing, right? We computed entropic scales, and then we trained a layered restricted Boltzmann machine on the adiabatic entropic coupling constraint, and this is what we got. So here was a sentence that came out when we asked the thing to create a sentence. The knave shook his head mournfully. Okay, that was a pretty good sentence. Sounds like Jane Austen. There's no ground truth. We have no idea whether one would actually say this is Jane Austen. But I think if anyone would look, they would be like, okay, sure, why not? The other thing worth noting is the sequence energy, right? So in current, you use ChatGPT or use anything else, Anthropic, Claude, whatever it may be. You don't know why the thing was created the way it was created. Here, the sequence energy actually is directly from both the parts, which are each one of these words, and the interactions between the parts, right? Similarly to the protein case, the knave shook his head mournfully retains grammatic structure in a way that has to have higher order correlations in it. This knows nothing about grammatic structure, right? So yeah, go ahead.
They're governed by this law, right?
Yeah, yeah, yeah.
That is this word, right, has like a clear hierarchical structure.
But the matrix does not.
Okay, but it's embedded from the.
Yeah, of course. Yeah, of course, right? What you say is not surprising is surprising to many people who don't, like it just depends on your point of view. I think that the fact that Zipf's law occurs and there's intrinsic hierarchy probably does mean this would work, right? Well, I don't know what less clear hierarchy is. So here's my thing on this. I had no clue this would work. Okay, I think that I now see it and it could work. It was entirely not obvious to me that 300 sentences by 120 words would get you something that would retain a hierarchy. The flip side is true in that we know of models that take hierarchical systems and destroy hierarchy, right? But to a priori say whether something is hierarchical or not, and then come out with it saying it's hierarchical and then be surprised, this presumes that we're very confident that it is not hierarchical to begin with. So it's hard to say. Have we done anything without a hierarchy? I don't know, right? I mean, like, I am not sure, and I think this actually gets to the point I'm trying to make. I am not sure that any of these emergent systems are not hierarchical. Right? I think this is the point. So why could something have worked on proteins, on MNIST, and on language, where, yeah, everyone, I mean, I don't think any protein biophysicist that I've talked to has said that the protein language models that Baker has created would work on language. This is not an obvious translation, right? But the same math that we've used for proteins does work on language and MNIST. Why? Okay. We think that there is a theory of emergence that underlies all of this that is intrinsically hierarchical. And I don't think it would work on if we knew things were not hierarchical. I don't think this would work. I think that is a completely correct point. I just don't know how to select for something that's not hierarchical, but yeah. What is this equation? Let's say you have a bunch of, let's say, recipes that exist, meals that exist in the wild, and they are made of ingredients. The idea behind this equation is to say the total number of states that you could effectively see is not random, it's not all of them, it's omega, and that omega is equal to the product of the probability distributions that are inferred at one layer and then dependent on only the layer before it. If you daisy chain all of those together, which is why you take the product from I equals 1 to n, where I is the index over the amount of scales of variance that you infer, you get the number of possible states that you could get from the parts list that you have. This is the equation that we want to test more generally to see, is this truly representing hierarchical statistical emergence that we would infer from what exists through a generative process that we have no understanding of. And to us, that actually does define what a lot of emergence is. They come from generative processes. These systems come from generative processes that are iterative in nature, whether those iterations are through evolution or through sociology. So what we're working on now is the following to round out this whole story. The first is that the layered restricted Boltzmann machine is entirely interpretable, meaning that you can go into any one of these layers and understand what the JIJ is. And you can see energies of systems as you move through the network. So the question is, can we visualize the construction of these emergent systems from the bottom to the top? I think this is just a matter of visualization. I think we can. And I think the second thing is, can we fix bad systems? So you could imagine writing a really bad sentence, like a non-Jane Austen sentence, and then using the layered restricted Boltzmann machine to now fix it in one-by-one ways. Same thing with a protein sequence, right? So we have data to suggest that there are protein sequences here that AlphaFold actually say will work, but they do not. How do you fix them? This is a very important problem because AlphaFold is not based off of any function. It's only based off of fold. And we know that fold does not equate to function. So could we use the layered restricted Boltzmann machine to now look at probability flows and create directed paths of mutation towards what the training data set would look like from an energetic standpoint? This would be in direct contrast to something like directed evolution, where you have no priors on what to create. You do random mutagenesis and selection. So as an alternative to non-constraint-based deep learning, what we think is that this could be a very useful way of going forward to think about how to create emergent systems leveraging constraints that are learned from statistics. Okay, so the last thing I'll end on is the following. A big question that comes out of this that we haven't really scratched the surface of, but this is for a future time, is what we've done so far is infer hierarchies from what exists. That does not mean that the true generative process is hierarchical. It may be something completely different. This is an outstanding question, I think. Is a layered hierarchy of entropy the true generative function of emergent complexity? And I'll point to a paper here from Herbert Simon, where if you guys haven't read this paper, I think it's great. It's about the architecture of complexity, and he goes through a parable of two watchmakers. And that's what all of this text is. But to summarize it, you have two watchmakers. You have one watchmaker who they both make the same kinds of watches, but one watchmaker has the following characteristic. They build it in ways that are hierarchically modular. So they build one module, another module, and then they take these modules and put them together to create a supermodule, and then they take the supermodules to create the watch. And then you have another guy who does not do things this way. Everything is piecemeal until you get the watch. And he makes the point that there's one of those guys that will win, especially when someone knocks on the door. So if someone knocks on the door, who's going to win? It's the person who can stop. Because if you need to create such a finely tuned thing piece by piece, then there's no obvious stopping point unless you have hierarchical modularity. So he says here, the watches that Aura made were no less complex than those of Tempest, but he had designed them so that he could put together subassemblies of about 10 elements each. 10 of these subassemblies, again, could be put together into a larger subassembly. Hence, when Aura had to put down a partly assembled watch in order to answer the phone, he only lost a small part of his work. And he assembled his watches in only a fraction of the man-hours it took Tempest. So his point is that hierarchical modularity is a great prior to try to think about how emergent systems can reliably come about without being intractably complex and therefore needing lots of time and uninterrupted man-hours, quote unquote, to create. Okay, with that, thanks to everyone in the lab. And we have several funders, but one of the funders is NITMB. All of this work was actually funded by NITMB. So we're particularly thankful to the organization for that. Thank you, guys. Yeah. Yeah, my PhD was with him. Yeah. So yes.
You're designing the course mate mutase.
Yeah.
Are you effectively? I know the protein correspond to the eigenspectrum of the something related to the MSA.
Yeah.
Are you effectively designing the protein sectors when you use the LRBM?
You are not actually. So the protein sector. So for those of you who don't know about this work, it's great work. So you take a multiple sequence alignment. You weight by conservation and then look for what is covarying with each other, and then perform eigendecomposition to take the top modes of variance. And the top mode there, or the top few modes, is what he calls the protein sector. That actually turns out to be the first layer of our layered restricted Boltzmann machine. So if you, roughly, right, if you create that first layer and you generate what you'll see is a high overlap between protein sector, phylogeny, and what we would generate. The key epistatic interactions, we think, are between layers. And the between layer interactions actually have to do, in the lingo of sector or non-sector, actually have to do with the progressive hierarchy from the sector to the non-sector. Yeah. Yeah.
Ideas. Thank you for sharing. So I guess like one of the original Desert data of entropy was this like conditional decomposition that like mutual information should be like, whatever you call mutual information should be a function that when you condition on probabilities has this like decomposition. And I guess the only sort of like this seems deeply connected to what you're talking about. What I don't quite understand is like the specific independent properties, like the orthogonality between subsequent layers, which is demanded by something like SVD. Like that seems like a special case of this more general decomposition that you're kind of hinting at, or that Shannon, in desiring something like that, would have hinted at. So I'm saying, like, why does that work? Or like, do you actually not invoke that assumption of orthogonality between subsequent layers quite so strongly?
Yeah, it would be nice one day to have just like a knob that you turn without needing to instantiate, like as a prior, that layer 1 is distinct from layer 2, because I actually don't think that's probably the way it is with respect to if you had infinite sampling. I think that a lot of what we're seeing is that it is two things. The first is that we have to just put some criteria of a threshold to have layers such that we can enforce sparsity in the JIJ. I think the second thing is that in the limit of finite sampling, it's hard not to say that some of those layers are because of finite sampling. So at the very top of the layered restricted Boltzmann machine, what are we seeing? We're seeing like very fine-grained differences that effectively probably can't be disentangled too far from you just didn't sample enough. We're chronically undersampled in all of these, systems. So I hesitate to say that there's anything like fundamental about modules with respect to modules being hierarchically layered. We just don't have enough to say that yet. But from our perspective, we've done this for a practical, it's more practical than not. Yeah. Yeah. Yeah.
Can I ask just about the idea that things flow in one direction of the hierarchy?
Yeah.
So I'm not a biologist, so I can't think of any very convincing examples, but it seems to me that, in some cases you also have feedback from the coarse level to the fine level.
Yeah.
So for example, in a tissue, maybe due to the whole tissue properties, one cell will start producing some protein, which is a very fine-grained thing to do, but it's based on the properties of like the entire tissue.
Yeah,
so
possible.
I don't know if you can like extrapolate my thought to something more reasonable. But you think that the hierarchical structure is really the dominant feature, and like in terms of actual living systems, there's little feedback from the coarse to the fine?
I'm not sure you would see that until you had dynamic sampling, right? Like the data, you'd have to have data which has that feedback in it that you were trying to.
Mm-hmm.
I think one of the things that you're kind of confusing is the statistical hierarchy that we talk about and the actual coarse to fine things that happen in a cell.
But isn't the idea to infer the actual like mechanistic hierarchy from the statistical hierarchy?
That comes downstream once you generate the samples, right? But in order to construct the generator itself, all we talk about are statistical hierarchies. And like she said, unless we have dynamic sampling where we see that event so often, we will not be able to resolve that kind of detail.
Okay.
But if you did that decomposition and you saw that event so often, it would be in the lowest layer.
Yes.
I mean, it's important also to understand that if you have a dynamical decomposition, if you had time series, let's say, the layer is not going to be, it's going to be entropic with a definition of entropy with respect to time in that case. So whatever is the lowest ordered thing, the most ordered thing is what you're going to see. So if that interaction between what is fast fluctuating and slow fluctuating is conserved and is really important for
It
the
will become slow.
then it'll become slow entropy. I mean, slow fluctuating in the entropic sense. So my feeling is that it would just get reordered
Mhm.
in the statistics of the ensemble. Okay, cool. Thanks, guys.